This video is on mRNA processing, which is how we take the pre-mRNA that we made during transcription and turn it into mature mRNA. The last video was about transcription, where we've made our pre-mRNA. Our pre-mRNA is made out of a sequence of nucleotides, that was copied from the template strand of the DNA. So in transcription, we read the DNA with RNA polymerase, and we made a complementary piece of pre-mRNA. So here's my pre-mRNA. That was copied from the DNA. So this is my pre-mRNA, which is my product of transcription. This video will cover how we turn the pre-mRNA into mature mRNA. This step of protein synthesis is called mRNA processing. mRNA processing has three main components. First of all is splicing. Splicing is when you cut out pieces of the mRNA that you don't need, You cut out what's called the introns. And in a second, we're going to discuss alternative splicing, which is a way to increase the diversity of the mRNAs you produce. Another piece of mRNA processing is adding a 5' cap. This is for protection, and so that the cell, the ribosome knows which side the mRNA it should attach to for translation. And adding a poly A tail, which is also for protection. So let's go through the steps that occur during mRNA processing. Like I said before, the pre-mRNA, also called the mRNA primary transcript, is the product of transcription. Within this pre-mRNA are sections of nucleotides called exons and introns. So let's go ahead and call, these first 6 nucleotides, we'll call this exon 1. And then we'll call the next, set of nucleotides here intron 1. And then right here, we'll call this set of nucleotides exon 2. And then we'll call this set of nucleotides intron 2. And we'll call this last set of nucleotides exon 3. Basically, what happens here is some of these exons and introns are not needed. So what happens is a piece of, it's called a spliceosome. A spliceosome is a complex made up of both proteins and RNA. So this spliceosome is going to come along. It's made up of SNRs, which are RNA and protein. The spliceosome is going to come along, and it's going to cut out the introns. And this is always confusing for students. Why do the introns get cut out and the exons stay? Well, exon actually stands for exogenous sequence, and intron stands for intervening sequence, which we want to get rid of. So what's going to happen is this spliceosome is going to come along, and it's actually going to fold up the exons so that they're close to each other. So here's exon 1, here's exon 2. It's going to fold the exons so that they are right next to each other. It's going to reattach those exons together, and that intron, which kind of makes a horseshoe in the back, it's going to be cut out. So this spliceosome is going to go down my pre-mRNA, and it's going to cut out my introns, and it's going to glue my exons together. So now in my mature mRNA, I'm going to have exon 1 right here. Glued to exon 2, glued, and by glued I mean covalently bonded, to exon 3. So in my mature mRNA, I've gotten rid of my introns. Okay. I'm also going to attach what's called a 5' cap. A 5' cap is actually a modified GTP nucleotide. This cap will denote the beginning of the gene, so that right here, when this mRNA attaches to the ribosome, here's my 5' cap. Okay, and the ribosome knows it should attach to the 5' cap side of the ribosome. I'm sorry, the mRNA should attach to the ribosome at its 5' cap side. Okay. So now I have my mature mRNA. I've added my 5' cap to say, hey, this is the side of the mRNA that should attach to the ribosome. Also, it's for protection, because as soon as this mRNA leaves the nucleus, there'll be enzymes that try to come and break it down. Then at the end of my mRNA, I'll add a length of A nucleotides, and I call these A nucleotides the poly-A tail, or we call them the poly-A tail. The poly-A tail is a group of A nucleotides that protects this mRNA from being broken down by enzymes. Because again, as soon as this mRNA leaves the nucleus, it starts being broken down. This is actually a form of control. The longer the poly-A tail, the longer the mRNA can exist, the longer it can be read to make proteins. So we can kind of like give a shelf life to our mature mRNA based on how long the poly-A tail is. Okay. So here is my mature mRNA. Here is a product. Here's my product of mRNA processing. After mRNA processing, this mature mRNA can leave the nucleus through the nuclear pores. It can attach to the ribosome and begin translation. But I want to take a second and really look at this step of splicing, because splicing is a really cool thing, and it leads to a lot of amazing adaptations that we use in our immune system and things like that. So let's flip our big sheet over for a second, and let's take a deeper dive into mRNA processing and splicing. Okay, so when I think about splicing, this is what I was showing you earlier. Here's exon 1, here's exon 2, here's an intron in between, right? The spliceosome takes exon 1 and exon 2 and puts them close together and cuts out the intron so that exon 1 is attached to exon 2. Okay. There are some evolutionary advantages to this. One, mutations in the intron don't usually affect the protein. Also, we have something called alternative splicing. This can help with the diversity of the mature mRNAs produced. So alternative splicing is exactly what its name implies. It is splicing the same pre-mRNA, the same pre-mRNA in different ways to produce different mature mRNAs, which can lead to different proteins. The body uses this methodology a lot when it's creating antibodies. Okay, so let's say that this right here is your pre-mRNA. Here's your pre-mRNA right here, okay? In my pre-mRNA, let's say this is exon 1. Let's say this is exon 2. And let's say that this is exon 3. And they're separated by a number of introns. Again, if there are mutations in the introns and they're cut out, it doesn't affect the protein. Let's say the first time the spliceosome splices this, they attach exon 1 to exon 2 to exon 3. And this is read to make a specific protein, right? The protein that's made maybe has amino acids that were coded for in exon 1, attached to amino acids that were coded for in exon 2, attached to amino acids that were coded for in exon 3, and there's your final protein. But what if instead when the spliceosome was cutting out the introns, it also cut out exon 2, which is a probability. So then you have exon 1 glued to exon 3. This can happen. You can't rearrange the order, but they can be glued together. Like you can cut out other exons as introns. So now the protein that's going to be made here is going to be exon 1, the amino acids coded for in the mRNA in exon 1, and the amino acids that are coded for in the nucleotide sequence in exon 3 are going to make the mature protein here. So you can actually make different mRNAs, which can code for different proteins using the same pre-mRNA. So if we think about the evolutionary advantages of having introns, one, mutations in the introns don't affect the protein because they're cut out before they're read to produce the protein. So this is a protective mechanism. And two, this can create diversity, right? Because of alternative splicing. The same pre-mRNA can produce different mature mRNAs, and then those can code for different proteins. And this often is used for antibody production in our beta cells, our B cells, basically. Okay. So at the end of this step of mRNA processing, we have our mature mRNA, okay, which is the exons glued together with a 5' cap and poly A tail. The next step of this process is translation, which will be in the next video.